but but when we talk about bacteria, so we are interested in organic matter because bacteria are the main heterotrophs. They consume organic matter. Okay, whereas phytoplankton, primary producers, they produce the organic matter. Okay, so now let's look at the organic matter dynamics in the seas. Okay, because organic matter are important food for bacteria. So consumption of this organic matter or degradation of this organic matter actually recycles the nutrients back to the food web. Okay, as you've learned earlier, a lot of the degradation recycles back the N and P, okay, the nitrogen and the phosphorus, but not the silicate. But the N and nitrogen and phosphorus are the ones that are important to most of the primary producers in the seas. So when we looked at when we are looking at the dynamics of a component in the sea, okay, for example, organic matter in the sea, we like to understand the inputs and the outputs. Okay, what goes in, what contributes to its concentration, and what depletes its concentration. So for organic matter, you can have two sources. Okay, so these are the terms that we use: allochthonous and autochthonous. Okay, allochthonous means external. So the source is external. For example, from anthropogenic activities, from the coast, from the rivers, entering the sea. So these organic matter are allochthonous, okay, from outside going into the system. We can also have autochthonous, okay. These are sources that are recycled in situ, okay, or produced in situ. So mainly by the primary producers within the waters itself. So you have two main sources, autochthonous and allochthonous. Okay. What are the different kinds of forms of organic matter in the seas? You can have particulate and dissolved. Okay, so as I mentioned earlier, when you talk about particulate or dissolved, we need to have arbitrary filter size to separate them. Okay, you can either use 0.7 micron, use a suitable glass fiber filter, GFF, or 0.45 micron filter, uh, uh, millipore nitrocellulose filter, or 0.2 micron filter. It depends on the filter that you use. Okay, so it is dependent upon the researcher what kind of filter they use, but you can see from literature, I either of these three is okay. Okay, some researchers will use 0.7, some researchers use 0.45, some researchers use 0.2. So for my lab, we prefer 0.7 because glass fiber filter is easier to clean. When you're working with organic matter, you need filters that are clean of organic matter. You do not want your filters to contribute further to the organic matter concentration in your sample. So glass fiber filter, you can bake it in the oven. You can bake it for at about 500 degrees Celsius for 3 hours to remove all organic matter in the filter. Then you can be assured that when you use the filter, you will not be contributing additional organic matter to your sample. That is why we choose 0.7 micron glass fiber filter. But other labs will choose differently. If you use 0.45 micron, you'll need to soak it in a mild acid solution to remove possible organic matter. Okay, so yeah, different approaches to cleaning your filters before you use them. So the particulate organic matter can be biotic or abiotic. So abiotic, non-living. Biotic is living. Okay, so for abiotic, it's like sand, silt, or clay. So for biotic, usually it's the larger organisms that are trapped on the filter. So particulate organic matter are those that are trapped on the filter. Those that the filtrate that passes through the filter are dissolved organic matter. Okay. Then for dissolved organic matter, you can further divide them into high molecular weight or low molecular weight. Usually we use Daltons to separate between high molecular weight and low molecular weight. So here you can see the organic matter pool. You will have a locturnous source, an external one, and the autochthonous source, either recycled or within the waters itself from primary production. Okay, you can have separation. This is how you can divide your organic matter: dissolved, particulate. Okay, so within the high molecular weight, you can have refractory or labile forms. But even in the low molecular weight, you can have refractory and labile forms. Okay, so for high molecular weight, for refractory forms, usually the humic acids and the fulvic acids, and labile are your cellulose. For low molecular weight, the labile forms are your glucose, carbohydrates, amino acids, nucleic acids, and all that. Refractory are usually the compounds that are already broken down by the bacteria until it could no longer be broken down. Okay, so it becomes refractory. So this is from Menzel and Reiter in 1968. So they carried out analysis of DOC throughout a water column. You can see they sample all the way to 5,000 meter depth. Okay, and they sample several times in a day, and this is the variation that they observe. Okay, the line is the average. The arrows here show the range of the concentrations that they observe. So this is an important uh, figure because it shows how you can interpret data from accurate measurements of DOC. DOC stands for dissolved organic carbon. So for DOC, you also have DON, dissolved organic nitrogen. So several things you have to see here, several statements that you can make. Initially, the DOC concentration in the upper waters, the DOC concentration is high, but in the deeper waters, it becomes low and quite consistent. Okay, and another observation that they made was in the upper waters, the variability is high. Okay, the lower range and upper range at each level extends over a wider margin as opposed to the deeper waters. So from here, they extrapolated that the wide the wider fluctuations that you see in the upper waters is because of the labile portion of the DOC that is being produced by primary producers and recyclers. So the autochthonous production of the DOC there's labile. Okay, you quickly utilized by the heterotrophs. That's why you see this wide range here. So from there, they extrapolated that these are the labile portion. Okay. So when they looked at the deeper waters up to 5,000 meter deep, they said it doesn't change much anymore. Okay, so they concluded that these are your refractory, non-labile or refractory portion because they are no longer changing. So they extrapolated it all the way up here to show that these are the refractory portion. So this, if this is the labile portion, this is the refractory portion. So this, they term it as semi-labile. So that's how Menzel and Reiter were able to categorize the different types of DOC within a water column. So from here all the way up, okay, up to 30 micromolar is your refractory portion. And from here to here, about 40 micromolar is your labile. Okay, so from another 30 micromolar in the middle here, that is semi-labile. So that's how you can use this to uh, interpret your DOC data. So measuring DOC is quite controversial in the beginning. Okay, so initially wet chemical oxidation was used, persulfate and dichromate solutions, where all the carbon in the seawater sample was oxidized into CO2, and then the CO2 is measured with a non-dispersive infrared analyzer. And later, a new approach, high temperature catalytic oxidation, was proposed, where the waters are heated up to 1000 degrees Celsius to oxidize any carbon into CO2. So this is the high temperature catalytic oxidation, first commercialized by Shimatsu, okay, Japan. Okay, so a bit of a controversy here. At the time when the company made this, so they had Japanese researchers testing it out, Sugimura and Suzuki. So they went to Pacific Ocean cruise. So Japanese researchers have annual cruises from North Pacific until South Pacific. So they have annual cruises. It covers the whole of the Pacific Ocean. So they measured the DOC concentrations along the Pacific Ocean. So when they're first using it, they measured it and they got very high results. The DOC was very high. So they were very excited. And they published it. This is around 1984. So they were very excited. They published it. Then they said, if you use this new approach, the concentrations of DOC are much higher. That means we have been underestimating the DOC in the sea. Okay, because that will have implications towards carbon modeling. Okay, because the amount of carbon that they are proposing is higher, much higher than what was previously known. So of course, for when something like this is proposed, other researchers bought the equipments and started doing the same experiments. And they found that they can't repeat the experiments. They are not able to get the same results. So something is not right. Okay. So something is not right. Uh, so they had a workshop where they did intercalibration between labs. Okay. In order to find out the problem. Then they found out that when you use high temperature catalytic oxidation approaches, when you use this kind of methods, blank is very, 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 very important. Okay. Because the blank can substantially change your results. So after the workshop, they proposed only to use ultra pure water to run as blanks uh, for Shimatsu TOC TON analysis. So Suzuki and Sugimura gracefully retracted their paper. So they admit it was there was something wrong with it, and they retracted the paper. So this is sometimes in science this happens when you're proposing something new, and over time uh, they found out that it's wrong. You can retract your paper. Okay, it's quite common. It's quite uh normal to do these things. Okay.